ladies and gentlemen, welcome back. So we are going to pick up today talking about the genomics of bacterial cells, so their genome, their DNA. So when we look at the major organization of a bacterial cell's genetic information, um, prokaryotes have a single chromosome. So take that double helix and basically bend it back on itself into a big ring. And it's a pretty big one. So if you look at it, here's a bacterial cell that is basically kind of dying in its process and just spewing its chromosome, its genetic material out into its environment. Um, and all of this is one continual chromosome that has been bent and wrapped back on itself in order to fit into the internals of that cell. Um, that being said, if we compare the amount of genetic material to a prokaryote versus a eukaryote, it's considerably less, has considerably fewer associated proteins that it can produce, and it is found in that generalized nucleoid region as opposed to eukaryotic cells being within a nucleus. Um, Most eukaryotic cells will have multiple chromosomes. The other major difference is that eukaryotic cells will also have linear chromosomes. So when you typically are used to seeing those X structures in things like mitosis and meiosis, when we talk about chromosome structure, you're talking about distinct ends of that bacteria. So there is a beginning and an end, if you will, as opposed to a big loop that we see here in bacteria. Um, There are a few other things that we'll get into later this year in terms of things that can attach to these chromosomes and help with their internal organization that are different, things that bacteria will lack that eukaryotes will have, so on and so forth. But we'll get into more of that later this year. So for now, genomic structure, right? Bacterial cells have a single chromosome and it's circular. Um, They also can have these accessory sets of genetic information in these rings called plasmids, and this is something that eukaryotes don't exist. And I kind of like to think of these extra plasmids as sort of like superpowers, because they'll contain a small handful of accessory genes. So it allows that bacterial cell who is carrying that plasmid to have some additional genetic information to do some additional processes or build some additional proteins that are not part of their main chromosome. So it's like extra features. And so it can be things like the ability to resist certain antibiotics. It can be the ability to metabolize um, nutrients that they don't typically encounter, so on and so forth. Cool thing about plasmids is that they do replicate independently of the main chromosome. So even if that bacterial cell isn't actively going through cell division, going through binary fission to reproduce, they can still make copies of these plasmids and they can also share them. These plasmids can be shared to the environment and then picked up by bacteria that are coming across it. They can also be directly shared from one cell to the next via conjugation, and we're going to talk about some of those processes then. Something you do need to realize, though, is that it does take energy for these cells to hang on to these plasmids and maintain them and use them. So if they get themselves into a situation where it's no longer beneficial to have that extra genome, they won't waste the energy keeping it, and they'll just expel it from their cell and move on. But if it is something that is providing them with some additional ability to survive, then they are more likely to keep them. So prokaryotes have a considerable amount of genetic variation, which is kind of interesting when you think about the fact that they are asexually reproducing organisms. So there's going to be three major ways that contribute to their genetic diversity, which is probably what has allowed them to survive since life began. So let's talk through these ones. First and foremost, they just reproduce really quickly. Um, Assuming that they've got favorable conditions in their environment, they can have some really short generation times. The vast majority of species that we are aware of, under favorable conditions, can divide and create a new generation anywhere between 1 to 3 hours. Um, Something like E. coli, under the optimal conditions, best conditions possible, can actually form a new generation in 20 minutes. Um, and so just having that ability to reproduce so quickly is going to allow the spread of favorable genetics that much faster. And again, keep in mind we're talking about human timetables here. So having that next generation in 1 to 3 hours is incredibly quick to us. Um, but it's just the way that they reproduce. They do divide by binary fission, so they are asexually reproducing organisms. They have no nucleus, so it's not true mitosis. There's no need to get rid of um, the nucleus. There's no mitotic spindles. There's nothing like that. Essentially, the DNA just makes a copy of itself. It goes through DNA replication, and in the process of that DNA replication process, it's pushing those separate copies of DNA to either end of the cell, triggers the cell to elongate. The cell goes through cytokinesis, splits, and you get two daughter cells. And they are genetically identical to each other. They are also genetically identical to the original parent cell. So they are all clones of each other. So keep that in mind when we talk about the amount of genetic variation that exists in these species, because there's got to be other mechanisms than just reproduction that allows them to get all of the diversity that we see, because they are just making photocopies of themselves. So the genetic diversity has to come from something other than just the replication, because they're not mixing up genetic information in the process of reproducing like a sexually reproducing organism would do. So where does the diversity come from then? Well, one of the ways that they can do this is, just like any cell, they can mutate. Changes in their DNA can happen as accidents of mistakes in cell division in that DNA replication process. Um, Again, I said keep in mind when we talk about rapid reproduction, we're talking about human scales here. If we divide it out, if we sort of scale it down and look at reproduction rates based on, or I should say mutation rates based on reproductive rate, the mutation rate is relatively low via binary fission, just like we would expect that to be in any cell, because these guys have evolved ways to ensure that they are reproducing accurately. But the difference here is that when those mutations occur and they're beneficial, they can really accumulate rapidly within the population because, again, their generation times are so quick on our time scales. So that high diversity from mutations is allowed them to rapidly evolve, mostly because we're talking about evolution and reproduction on our scale of time. But again, if we were to scale this all back out, it's really not happening any quicker or at any higher rate than we're seeing in more complex organisms.